So after PCR is done, you will get your samples back. Hopefully they will be thawed, but on ice. You may want to tap the tubes down if there appears to be any liquid on the sidewall. And then we want to run gel electrophoresis on these samples. This will separate your fragments of DNA by size. But first, in order to get this sample to sink in our liquid, and so we can track the DNA as it goes down our gel, we've got to add loading dye. This loading dye here is 10x. I've seen at this campus 5x and 6x. So check your concentration, and you want to get it down to 1x with your sample. Since this is 10x, my volume in here would be 40 microliters. So we would add 4.44 microliters to it. If it's 5x, it would be a different calculation. So I got my 4.44 microliters of DNA, or not DNA, loading buffer, and I'm going to add it to my DNA. And you want to pipette this up and down in order to mix. You can also gently flick the tube if you prefer to do it that way. But again, you want your sample to be homogeneous. The whole thing should look blue. I'm going to still use this 10 microliter pipette to load the gel. DNA is negatively charged, so it will go towards the red cathode. So you want to have your wells opposite the red side. So I'm going to actually flip the box around. So your wells are going to be next to the negative side, the black side. And they're going to go towards the red side. So this is how you want your gel in the gel electrophoresis box. And now we're going to load my sample. We do have loading tips. You could do this volume all at once with a P200. But for this, I'm just going to use the P10 a couple times. So in here we have our wells. I'm going to put my pipette tip under the liquid. I'm going to insert it into the well, and I'm going to move my pipette tip back and forth. That way I can feel the sides of this well, and I know I'm actually inside it. And then I'm going to depress the plunger, and you should see that well fill up with blue. Then I'm going to grab the rest of my sample and just do it again. Now, if you're doing it, whenever you do load, you want to make sure you don't overflow these wells. These wells should handle 50 microliters. So with our sample of 40 microliters plus our 4.44 loading dye, they should hold the entirety of your sample. However, it is going to get full at the end, so you want to be really careful and really slow with that plunger at the end. So that well is loaded. I'm going to load some loading dye next to it because the edges tend to smile or curl up whenever they run. So I'm just going to put 10 microliters of loading dye right there. And then we'll put the lid on, and I'll show you how it looks when it's running. So when you put the lid on, make sure you're color coding this. Red should be with red, black should be with black. And then you want to plug in color code again. I'm going to set this 140 volts is fine, and just press start. And you can tell there's a current running through it. At the ends of these boxes, you should see very tiny bubbles. It should look like very active champagne fizzing. So when you see those bubbles, you do know your gel is working. And over time, you should see your blue dye travel down your gel towards the red. If you ever see your blue dye start to go back up, you've got it backwards. As long as the dye hasn't gone off the gel yet, you can still stop the machine, flip your gel around, and restart. But if you make sure your wells are closest to the black end and opposite the red end, the gel is going to go the correct way. The gel should take 30 to 45 minutes once you get it loaded. And after the gel is run, then you can stain your gel and see results the next day.